afternoon presentation on the breakout. I'm not going to go through the really basics of soil health. So if you want to know that, there is Soil Biology 101 on my community, which is at jillclapperton.com. And that's all recorded. So if you want to know something more about that, then you can go there. And if you want to ask me questions, my email is here. You're welcome to do that. And I will get back to you. So, and also there's more information on our website too. So, you know, if you're interested, there's some stuff there too. Okay, so we'll get started here. And the first thing is that this is a model for regenerative agriculture. And I see this as being the new conventional. We're all going to be, you know, thinking a whole lot more about what we put into the soil, how we manage the soil, all of those things, especially with fertilizer prices the way they are. We have to think a lot more about how we can use biology to get where we need to go. And the biological properties of the soil are what really unite the chemical and the physical properties of the soil. And together, all of that soil health then is a measure of soil productivity, affects soil productivity. And as we measure soil productivity, I mean, what do? Most of the time, we are measuring soil productivity in terms of yield. Well, I'd like us to more measure it in terms of food quality, because I think ultimately we should get paid more for quality food that we're producing. And some of the environmental benefits that people are getting from farmers and who are managing things in this way, because we're not putting more soil into the environment. We're not, you know, sedimenting waterways. We'll have more water use efficiency, which is a big deal because we all know that our aquifers are slowly drying up. And ultimately, we'll have better health for ourselves and for our animals. So the first thing is we have to have created a habitat. And the way we do that is going to be with no-till. And the reason I say that, you can use conservation tillage too, but no-till is always going to work better. And that's because once you create a habitat, or once you have infrastructure, if somebody came around twice a year or once a year and destroyed all your infrastructure, you know, that takes a lot of energy to rebuild it. And soils have an infrastructure. They have pathways. They have continuous soil pore network that allows all the protozoa and the nematodes in that to move around in the soil so that they can control the levels of bacteria and fungi so that plants can actually get organic nutrients out of the soil. So it's really important that we maintain and build on that habitat. And one of the ways we can do that is with roots. Actually, it's probably the best way we can do it is with roots. Lots and lots and lots of roots. This is a cover crop experiment in Germany. And this is the biggest soil pit I can honestly say I've ever been in. There were 300 people plus at this field day, and they were all moving through, looking at roots, looking at the different covers. You can see them all here. It was impressive. But what was also really good, it was deep enough that we could actually, and you can see this pit was probably somewhere around 6 feet deep, maybe deeper. And we had roots right to the bottom of that pit, and we had lots of them. And that's what's really important, because if we're going to build soils and we're going to build water holding capacity and nutrient recycling capacity, we have to fill the entire profile with roots so that we build a profile of soil, a really deep profile that has a lot of infrastructure so we can capture every bit of water that hits the ground. And this is from the Nature Conservancy. And you can see that, you know, if you're not doing no-till, the chances of you building up soil carbon are not that good. And we really need to be able to hold the water. I mean, that's what we really want to do right now. We need to be having increased water holding capacity, which will have increased nutrient cycling capacity and all of that. This is a really old picture. But you can see that when you add grass or forage into a corn rotation with corn, you actually start to build soil carbon. And part of that is because when you rotate with legumes, the legumes add a lot of nitrogen and you actually like use a lot of the carbon and you don't accumulate it. So this is about finding a balance between carbon. Now this is more about residues, even though it's carbon levels in the soil. If we had more roots, and part of this is because there's roots in the soil and more roots in the soil when we grow forage, which means that we actually build carbon, and that's the only way we can do it. Carbon is a function of plants, and we have to be growing a lot of plants in the soil if we're going to build carbon and really take advantage of the credits. And we want deep roots, because if we can get more deep roots and fill up the profile with roots, then we're going to build carbon in the soils a whole lot faster. And the benefit isn't just carbon credits, payment for carbon credits. It's a lot about water holding capacity and nutrient recycling. So increasing the quality and the quantity of the soil organic matter will grow populations of soil microbes. It increases your water infiltration and holding capacity. You get improved soil structure, which means that you have better predator-prey relationships. And like I said, it's not just about the quantity, because the quality is really important too. That's why we need to put brassicas and legumes and cereals all together when we do this, because they all are contributing different biological properties to the soil. So for example, if you when you add brassicas, the brassicas really build up the soil biota. So the soil animals love brassica residues. They love brassica roots. They love everything about brassicas. And the legumes have a lot of enzyme capacity that really stimulates a whole bunch more biological activity and the recycling of carbon, so that we're actually releasing nitrogen and phosphorus and whatnot from the organic matter. So you need all of these things together. And I think that for some reason, I seem to be skipping over slides. So just give me a second here while I make sure that I'm not skipping over things. Each plant species contributes its own signature to the soil, and it modifies the soil to suit its own purposes. And crops will actually do a lot of manipulation to the soil. So for example, if a seedling isn't getting what it needs, or it's a little too salty, it will leak out a lot of water into the soil to dilute the solution. If plants aren't getting enough nutrition from the seed, then they'll actually give off other things so they can trade them for what they really need. And ultimately, that's why seed quality is so important, because a seedling should be able to live just off the resources of the seed for 10 to 14 days. And if it can't, then we know that our seed quality is really poor and we need to deal with that. So the rhizosphere, and again, I'm just going to check that we're getting the right ones. The rhizosphere is the plants, soil organisms, and the soils all together. So it's the plant roots drive everything that goes on in the soil. Plant roots define the organisms that live in the soil, the plant microbial community that's in the soil. They define the soil structure. So roots can actually make the soil structure more crumbly, or they can make it more aggregated depending on what they need. If they need things to be more consolidated for purposes of water, then they will make sure that the soil is really attached to the roots, and they'll be actually trying to glue it together through the use of mycorrhiza, or just by exuding a lot of carbon into the root system, so that will attract a lot of bacteria that will actually bind the soil to the roots so that they can have access to more nutrients. So plants aren't being passive here. All of this is very dynamic. And there are a bunch of beneficial interactions. So for example, corn. If corn is being fed on by corn rootworms, root borers, any of that, they actually send off terpene signals. These are terpenes, sesquiterpenes or a beta-carboxyphylline. And what it is it actually attracts insect pathogenic nematodes. So that actually will, attack the insect larvae and the pests that are actually attacking the roots. So again, plants aren't being passive here. This is another reason why we need to feed them well so that they can do this. Lupins are another example. We can put lupins with corn and they will burst a lot of organic acids to release phosphorus if the concentration gets too low. Lupins can be very good at breaking down glyphosate, for example. As long as you haven't overused phosphorus, and it's just slightly on the low side, lupins will just make so much more phosphorus available. And mycorrhizal fungi, not only do they increase the rate of photosynthesis, they increase the movement of carbohydrates, amino acids, and organic acids to the roots, and they're trading for mineral nutrients and water with the plant. And because of that, they actually determine the microbial community that's in and around the roots. And I think what's important here to remember is we talk a lot about carbon. Root exudates, yes, a lot of carbon. Amino acids also have a carbon backbone. Organic acids have a carbon backbone. Proteins have a carbon backbone. So all of these are sources of carbon. And it just turns out that amino acids and organic acids are a more complex form of carbon, but they also have phosphorus and nitrogen and other elements attached to them that microbes need. So it's really important that we get amino acids and organic acids into the rhizosphere to drive, again, that quality of carbon. And you can see that it's water resistant too. So what that means is that you can see how the mycorrhiza, so fungi that actually live in the roots of plants, actually is holding the soil around right next to the root so that it can actually get access to the mineral nutrients and water. And then if we look in this slide here, you can see the glue, the glomalin, glowing. So once again, it's glued everything around the root so that it's actually having access to water and nutrients, even if it's really dry. And this is another way you can see building soil structure. Fungi are really important for building structure. And soil structure, that infrastructure, continuous soil pore network, is what really is the top characteristic of soil health. Healthy soil has good structure, excellent structure, allows for good water infiltration, holds water so that all the microbes can keep going all the time. Because remember, your soil is like this big digester. And the mycorrhizal hyphae, which are all these fine threads here, are actually holding the soil together. So they're binding it all together, making bigger aggregates. And once again, to benefit the plant, because the plant needs to have that soil around it so it can actually pull things out of it. And this is knapweed. So anybody who's grazing might know knapweed. Most of our weeds are highly mycorrhizal because they're taking advantage of the mycorrhizal fungi. So they're actually using their network, not only to aggregate the soil, but also to bring in nutrients. Part of that's the fact that weeds haven't been overbred. They're not used to getting any fertilizer or anything like that. So they're going to take advantage of nature's symbionts and actually use them. These structures here and here are actually where the plant cell is interacting with the fungi and they're carbon trading. So mycorrhiza were the first carbon traders. I mean, they trade mineral nutrients and water for carbon substrates from the plant. And we see here all different seeds. The importance of this is that every different seed you put in the ground, every different one, creates its own rhizosphere. And it creates its own microbial community around it. So legumes, for example, will make sure that they get rhizobium. they'll get the bacteria that they need in order to control things. So for example, they'll bring bacillus in if they're having some problems with insects as well, or other diseases, because they produce a lot of antibiotics. In a talk I was just doing before this, we were talking about potatoes and, and scab, and how important bacilli are for controlling scab on potatoes. So you want to have a microbial community that's encircling your plant roots that keeps other things away. So a beneficial rhizosphere. And each plant is trying desperately to do that. So this gives you a little example of the rhizosphere. In this case, this is faba beans, this is soybeans, and this is corn. And you see the differences in the pH. What's really cool about the soybean one is you can see where all the nodules are. That's where the bright yellow is, where it's more acidic. And then you can see the tips where it doesn't have nodules, where it's more basic. And that's another reason why mixing plants is so important, because then we don't get into these problems where we have very alkali soils and we keep them there, or very acidic soils and we keep them there. We need to mix and match so that we're always trying to stay in that neutral zone, or in that 6 to 6.5 zone. 5.5 is okay too, but we want to stay in that zone. And this is another reason why, like when we're building carbon in the soils, we're building roots in the soil, because we're talking here about creating really healthy soils. We have to put more roots in. So this is work from Green Cover Seed. And you can see these are soybeans at the same period of time as this 12 species mix was in. This is the drawdown. And you can see that we have the same parameters on there, same time. And look at the drawdown of the soybeans compared to the mix. And a lot of people look at this and go, well, that can't be right. I can only grow my cash crop because anything else just competes with my cash crop for water. Well, it's not true. Actually, plants will help each other within a community. So when you create a plant community, just like the prairie, it won't dry up as nearly as quickly as your cash crop will. Monocultures dry up because they haven't got anybody else to help them. If the corn starts failing, what other plants are there that have different root structure to help them? Who has more mycorrhiza? Who can do some of these things? They don't have any services. So the important thing is to actually think about, well, how can we grow companions between our corn rows? Is there anything we can grow with soybeans to also help it along? So let's explore that just a little bit. One of the other reasons that we want to keep mixes is because, so this is cation exchange capacity. So the soil releasing. So broadleaves, or dicots, forbs, whatever you want to call them, actually change the cation exchange capacity so that they're looking for things with divalent cations or anions. So things like calcium or sulfate or nitrates or anything like that that is a -2 or +2. Whereas your grasses are looking for more of your monovalent cations, which are potassium and sodium and chloride and some of these other ones. And when you mix them together, they actually get the whole picture, and you're actually tapping into the entire soil profile. And that's what we really want to do. And again, we mix them together, we can also mitigate the pH changes, and we can actually start, instead of mitigating, say we actually can facilitate a more beneficial pH change. And so this gives you an idea, like you can see that we've got some vetch there intermixed. And actually there's some really, really super yellow roots in here. It's the only way you can see mycorrhizal fungi without a microscope. If you don't have a microscope, you can't see mycorrhiza. And so, except with this yellow color. The only problem with the yellow color is the yellow color disappears really quickly in the sunlight. So you kind of have to break them open, look at them really quickly, not in the sun if you can, just to get an idea of whether you have mycorrhizas or not. But the important thing about this is you can see all the roots are woven together. they're sharing. I mean, we know that roots share. We know that plant communities are far more resistant to drought than monocultures. And again, there are some things we can do in a management system, a managed system. So I just want you to see some of the different legume root structures. They're all different. Persian clover is very resistant to drought. And you can see it has a really fibrous root system. Puts a lot of biomass actually in the soil. This one has a coarser root system. There's still a lot of biomass, fixes a ton of nitrogen. Sun hemp, depending on where you are, can get really quite big and produce a lot of nitrogen. And woolly pod vetch will just stay fairly prostrate. Doesn't give you a lot of root biomass, but it gives you very woody biomass that's resistant to decomposition. So all of these have their own properties, all of which we can actually use together. Sorry about that. Forgot to turn off my phone. I'm in my office. Huh. All right. So chickpeas and flax. This is kind of the no-brainer of intercropping. This works brilliantly. It's fairly easy to do. First thing it happens is that you don't end up using hardly any fungicides at all. And flax is a really valuable oil seed. Chickpeas are a very valuable protein source. It's not for everybody, but this is a great, if you're going to start even on a small scale, you just wanted to see how to do things, this is a great one to start. And chickpeas and flax are both very drought resistant, both tolerate the heat very, very well, but they like to be seeded early. So this is something that you could seed before corn, easily before corn. They like it cool to start with, and then they like it hot when they're flowering and when they're forming seeds. Sunflower, faba beans, and crimson clover. You can see how good the sunflowers look. You can see between the rows, there's 3 rows of faba beans, crimson clover is in there. This out-yielded anything in the area. And a bonus, there was a flush of faba beans after the sunflowers were harvested. And so there was a second crop. That wasn't the intention. The intention was just to feed the sunflowers. There's no fertilizer on these sunflowers. There was no disease. It was brilliant. And the only costs here were the cost of seed because, you know, didn't even spray for grassy weeds. Buckwheat, soybeans ripening together. This is a relay crop where you take off the buckwheat because the buckwheat will ripen in about 6 weeks, and then the soybeans come later. You get the benefit of all the bees in the buckwheat. You get the benefit of it holding up all your soybeans. This is in Iowa, and it grows really well together. And not using as much water as soybeans would normally. Once again, that weird thing. Wheat and lentils. If you are going to grow a cereal crop in your system, then growing them together is a good idea. And winter wheat and winter lentils grow really, really well together. And the lentils will feed the wheat. And again, we can cut back on fertilizer and still get yields, and very acceptable yields. This one sort of came about because for those of us that grow lentils in the Pacific Northwest, after we harvest and then we go into our winter wheat, oftentimes we have volunteer lentils everywhere, or volunteer chickpeas. And so we just seed directly into them and don't worry about them come spring. That's what it looks like. That was at harvest. So diversity is the key. We can reduce pests and pathogens. We are going to, you know, plants have a greater biochemical diversity than animals. Monoculture farming, yeah, we need to rethink our model there. Even with all the genetic manipulation, we just are setting ourselves up to fail because we're breeding pests, we're breeding weeds and all of those things when we do things over and over again. So the important thing is to mix it up, because when we mix it up, things don't, there's that sort of like hesitancy. So for example, with canola, when flea beetles and lygus beetles are flying over, they like to see a beautiful single color, because that tells them the resource is really good. If the color is a little mixed up because there's some grasses in there or there's something else in there, they will fly to a different field because it's like, that one's not a really good resource. The other thing, if everybody in the area did the same thing and they all had mixed crops, then the insects, then there wouldn't, no insects wouldn't, like, they'd be dispersed more because they'd have to go into smaller groups, which would be a whole lot better for all of us. And plant nutrition is really critical here, because if we're going to defend, if the plants are going to defend themselves, and if we are eating them, we also want to eat the secondary plant metabolites. They're also important for us. And micronutrients are the absolute key to plants actually producing these metabolites. If they don't have enough nutrition, especially micronutrients, they can't produce them, which means they can't defend themselves. And that's part of the reason why these mixed communities work, is because mycorrhizas can share micronutrients. Plants that don't need it at the time can share it with other plants, and then they can produce these secondary metabolites that allow for communications as well as defense. So for example, if an aphid lands on a leaf, what happens? Or, well, let's just say, so now I have a corn root borer, and it lands on the root, starts making a hole. Well, that root now starts to produce all these sesquiterpenes. So now it has to have all those micronutrients and the sulfur in order to do that. When it starts producing them, they all have an odor. Think of essential oils. Essential oils are all terpenes. So they have a smell. That smell is volatile, so it can move through the soil network. That's also why structure is really important, because then those chemicals can actually move, and the other plants can go, oh my, we're under attack, and they start producing them too. And the next thing you know, you've got a flood of nematodes coming in going, oh good, this is a good food source for us, and off we go. So you can see there's nothing, everything in the soil is really dynamic. It's not just a place where plants sit and we walk on. It's actually, there's a whole lot happening in there. And when it has a really good habitat and good soil structure, more can happen, more services can happen. We're going to have healthier plants. We're going to have a whole lot more activity going on below ground. If we use too much nitrogen, and nitrogen is always a balance, we have to find the balance with nitrogen, because we get too much nitrogen and we won't take up enough manganese and zinc. And if we don't take that up, enough manganese and zinc, our plants will succumb to infection. And that's kind of like us too. We also have the same problem. If we don't have enough manganese and zinc, we have the same issue. So just to give you an idea, you can see that all my roots in a monoculture are all the same. All my chemicals are the same that they're being exuded. When I have a multi-species cover crop, or I have some other crops or other plants in between the row, I can do different things. Now, and I get different molecules, I build the profile deeper. Now let's just say that we're going to do companions, because honestly, sometimes cover crops are really hard for people to do. Companions are a whole lot easier. So let's say we have companions. Well, if this is my crop, let's just pretend this is a corn crop, then I might want to grow these and these together, because they're going to be in a different zone rather than my major crop, my cash crop. So they'll be tapping nutrients and pulling up nutrients from a different zone, which means that they won't be competitive at all. And that's the kind of thing that we need to think about as we move forward in this. Durum wheat and faba beans. We were growing durum wheat and faba beans together. And you can see the faba beans had a way deeper root system than the wheat. They fed each other beautifully. I know this is in Saskatchewan, Canada, but Lana Shaw Group, the Southeastern Research Farm, is leading the way on companion planting. Corn likes multi-species covers too. So this is a corn that was grown on a previous cover crop. This is one that was grown, this was grown on a previous cover crop. This one didn't have a cover crop. And you can see the difference in the quality of the root system, the diameter of the stalk, all of it is a much better, it just looks better. You're getting more nutrients. I've got deeper, I'm tapping nutrients deeper into the soil, and I have more roots, which means that I'm also getting more minerals and nutrients out of the soil. And this is the same when we have companions. So this is companions in corn. In this case, we have pigeon peas, sorry. This is pigeon peas. And then this is what the pigeon pea looked like. You can see it doesn't have a lot of branching roots, but it has a very deep taproot. I really like the pigeon peas in this. In the end, this was the first of a number of experiments. In the end, and this was just in southern Iowa, in the end, what we did is we grew a mixture of legumes in between the rows of the corn so that we could actually put more nitrogen in and have a little bit more diversity. And it did not affect the crop at all. The crop was very healthy. And we were on dry land, we were yielding 300 bushels. So there's nothing wrong with that. We also had guar, we had mung beans. Like I said, we put things together. The guar really didn't like the canopy of the corn at all. You can see the mung beans, they were a lot happier. Well, actually, this is our first trial. And the first time you can see actually our yields were down 150, 200. We're up around 300 now. But this was the first year. This was about 5 years ago. And you can see these were all the trials we put in. And you can see what's interesting is like the three sisters, when we grew them together, they were doing quite fine. Some of the Czechs, sometimes not that much different from the Czechs, but there were some variations. So this is what the cobs look like. And you can see the cobs were filled right to the end. This is Roundup Ready corn. And you can see even though the sun hemp, some of them had good yields, they were sort of shorter, stubbier cobs, and actually had more cobs. And we kind of needed to get some of that research done. But this gives you an idea of what the cobs look like from that study. This is another one later on. And you can see we had companions in there. Look at the root structure, and you can see the stalks of the corn were much, much more robust. It didn't affect yields at all in this case. I mean, it was all fine. And still, we didn't have a water issue, but we were building way more soil carbon. We had a lot more roots woven together and sharing. This is studies that I've done with wheat. We had the cover crop the year before, and there were 15 different cover crops. And you can see that they were all seeded the same variety. You can see there were a lot of differences between yields, and there was a lot of differences between the nutrient density. Whatever you're growing beforehand has a real effect, or in the companions, can totally affect the uptake and a change in the nutrient density of the crop that you're growing as your cash crop. And part of that, again, is that one plant exuding more acids, bringing in more cations, more mycorrhizal, a crop that's more mycorrhizal, taking in more zinc, taking more boron, helping out the next crop. Plant communities tend to be just a lot stronger, and a lot, even though this is monoculture afterwards, having a mixed plant community beforehand can really affect your cash crop. So if you can get one in, fine. In the Pacific Northwest or in parts of, you know, the Great Plains, people are still doing fallow. So that would have been more of a fallow replacement. I find companions to be, or intercropping to be, a better way of doing things, because at least it fits with my climate better. I can't always get a cover crop in after I take my crop off. Half the time I don't have enough water, and then it gets too cold, and yeah, there we are. So a companion crop can actually allow me to get more diversity into my system. And then after I harvest my cash crop, I just let my companion crop go crazy. And then I have something covering the ground all year, like until I get back in the spring. And then if it dies over the winter, great. I can just seed back into it. If it's alive, I can still seed into it and, you know, deal with it after I've seeded. Genetics matter. And Michael Grusek is at North Dakota State University with the ARS, and they are doing a fabulous job right now of looking at pulse grains and how they're adapted to the prairies, and where is the mineral nutrient density, and where can we get the best yield and the biggest bang for our buck in terms of mineral nutrients, because legumes are an excellent source of nutrition for animals and people. And, you know, these are what we chose. And this is where the mean level of the nutrients was. Wouldn't it be better if we could have chosen up here somewhere so that we got a lot more iron and got a lot more zinc? Yeah, we need to balance, find that balance. This was Quinn Organics. This is a wheat variety trial. All these wheats were grown in exactly the same conditions, exactly the same time of year, in the same soil. And you can see all of them had different levels of various nutrients. One of the questions in the study was, well, everybody says that the old wheats are better than the new wheats. That's not really that true. The nutrient density of some of the new ones is excellent. The one thing that we learned was that none of them were really great on all nutrients. Some of them were better on others. And so probably blending varieties for flour or to consume was probably a better idea than just trying to grow one. And there's nothing wrong with doing that. So the rhizosphere is the most biologically active part of your soil. That row, that crop row, drives everything. It drives your carbon, it drives your soil structure, it drives everything. So if we can keep plants on growing all the time, we're gonna have more carbon in our soils, we're gonna have better soil structure, we're gonna take every bit of water that nature gives us and we're gonna hold on to it. And we're gonna have better nutrient cycling, so we can reduce our need for chemical fertilizers. We still probably have to supplement, but, you know, we get away with a little less. Once again, this just is about emphasizing the need for optimum plant nutrition to avoid diseases. Potassium deficiency really gives you a lot of high sugars and amino acids in the leaves, which attracts insects and diseases. But then you get too much potassium, and you're struggling to have calcium and magnesium and nitrogen. Nitrogen and potassium are totally antagonistic to each other. Too much potassium, not enough nitrogen. Too much nitrogen, not enough potassium. So it's really important to find the balance, really important to think about fertilizing your crop in crop as well. More small meals, checking leaf tissues analysis, making sure that you need more, and doing that before you need it. Calcium, boron, and zinc deficiencies really cause problems with leaky membranes, which causes more root nematodes, plant parasitic nematodes. It changes the concentration of photosynthesis in the leaves. And what's cool about this is that calcium, boron, and zinc move in what's called the water system. They don't move with like the sugars and stuff. They actually move up the plant. The plant draws them up through the process of transpiration. And if the roots get too hot, transpiration stops. So if we have something covering the roots, we have, let's say, a companion in there, keeping that soil really cool, we'll actually move a lot more calcium, boron, and zinc into the corn and into the upper levels of the plant. And again, plants need phenols in order to produce nasty compounds that prevent them from getting sick. And the only way to do this, they need micronutrients, and they need some N, not a lot of N, just enough N. And I think I've really stressed this. I mean, if we're going to keep our plants healthy, we need to have good nutrition. So let's move beyond that. We, in my view, and my world and global view, we don't have a food scarcity at all. We as farmers can feed the world, no problem. We need better distribution of the food, for sure. Because without it, we're going to struggle, but we need better infrastructure to distribute food. And if you want to control a population, what's the best way to do it? Starve them and corrupt their water supply. It's pretty simple. And that does happen in some countries. Zinc is really important, and I focus a lot on zinc because it has deficiency in zinc has been linked to Alzheimer's and Parkinson's. It's been linked to premature aging, causes inflammation. You have a lot more stress and inflammation if you don't have enough zinc. You certainly won't be, you'll be struggling to maintain, to fight diseases and whatnot if you don't have enough zinc. We know that you need to have a full complement of zinc and iron in order to have a really good outcome of orthopedic surgery. So anybody who's getting knees replaced, hip replaced, doing any kind of orthopedic surgery, need to make sure that you have enough iron and you have enough zinc in order to make sure that you have the best outcome possible. Plants are also important for pain relief. So let's get into the meat of this. Healthy bread that could save your life. So this bread was made with North Dakota and South Dakota wheat that they bought at $25 a bushel because it had high selenium, because the soils there are very high in selenium, which causes issues for livestock, but is really good for, you know, grains. And then it was blended with other wheat in order to get the right balance of selenium. Selenium being really important for reproductive health. But in the States, most of us are deficient in potassium, then vitamin D, vitamin E, vitamin K, magnesium, calcium, vitamin A, and vitamin C. And then of the micronutrients, zinc is the top one where we have deficiency. And grains of all kinds are the best source of potassium. And farmers are the ones who are supplying all these. The vitamin D, not so much, because we need to eat, you know, oily fishes to get most of our vitamin D, or just spend 15 minutes in the sun and get 25,000 units. This just tells you that most people aren't outside, which usually isn't a problem for farmers. Two corn varieties. This is X-ray fluorescence spectroscopy, which I've used a lot. I use this because it's handheld, because I don't need to have a fancy lab in order to do a lot of analysis. What this shows, this was a leaf off these two kinds of corn. And you can see the green one here is Pioneer. The red one is the DeKalb. And they totally differ in their composition. at this time. When I was at Missouri Corn Growers, all the farmers started laughing at me and said, yeah, well, we don't like to grow Pioneer because it falls over in the wind. Well, that would explain it. It has a really high silicon content and be very, very rigid. Same with the calcium. So structurally, Pioneer corn is really strong, whereas the DeKalb is not nearly as strong. But DeKalb has way more potassium in it, again, finding that balance. So the DeKalb probably needs more potassium. And in this case, the Pioneer would need to make sure that the calcium and the silicon is up in order for it to grow really well. So part of this analysis can also tell you what your particular variety needs. Comparison of mineral nutrient density in grains. In this case, all of these were grown in with companions or intercropping, but just gives you an idea that there's a lot of differences. Legumes, for the most part, will always have more phosphorus. They will usually have a lot more iron or more iron, and they will be high in zinc. Mustards are also one of those plants that actually are really nutrient dense. And these are just the grains. All we've been doing here is analyzing the grain. This gives you an idea of some different ones. This is red fife. This is David Brandt's farm in Ohio compared to a farm in Saskatchewan. And you can see there are differences. Some of that's just the soil. All soils have a carrying capacity, an innate carrying capacity. We can use soil, we can use fertilizer to push it a little bit, but generally speaking, it has a carrying capacity. And we're going to struggle at certain yields limits just because it's like that. We can increase that with increasing organic matter for sure. This is an old variety. This is Turkey Red. And once again, you can see the two differences. I couldn't do those nutrients early on, so this is what happened later on. New varieties, Glee, some, Kelsey, and then this is wheat grown with a cover crop, and then one with without one. And you can see that there are definitely differences when you have it. So just trying to show you the differences. This one down here, this is barber wheat, grown in 3 different states the same year. Okay, that would mean different conditions and all of that, but you can see there's still differences between some of the elements. Aluminum, no. Sodium, generally speaking, not. But some of the other ones, yes. And you can see the last one, 17 ppm of zinc, that's like way too low. But these could all be blended and you'd get a better mix. Right now, we're in the process of building a lab. We'll be able to do a lot more nutrition on some of the things we're growing. But I thought you'd like to see sort of a comparison between different grains. And you can see that this is another reason for having mixing species and whatnot, because not all of them do exactly the same thing in exactly the same way. Some of them take up more silicon. Some of them would return more silicon. Some of them take up more phosphorus than others. And again, these are partly from the soil, but they're also partially the way we grow them. This one was grown with, this barley crop was grown with companions. This one was not. Could that have made a difference? Yeah, probably did. This is for corn chips, and all of these, one of the varieties was grown in Mexico in all cases, and the rest of them were all grown in Ohio. And 4 actually corn chips, looking to create tasty corn chips that were different colors. I think it's really interesting here that if you look, so these are the red ones. This is the blue corn. This is the white corn. And then this is the yellow corn down here. You can see what's really interesting is some of the red corns had way more zinc than some of the others. All of them had just about the same amount of phosphorus, except for this one, the rosado. And the rosado was grown in Mexico, and it definitely didn't have nearly the amount of nutrient as the other. Bloody Butcher is one that's grown a lot in the United States. The blue corn, all very similar. The white corn, very similar. The yellow corn, mostly similar as well. So just to give you an idea of the corn, that you can have difference in nutrient density. All of them, with the exception of this blue one, and this rosado, would be excellent sources of potassium for people. Source of calcium, terrible. We want to see over 600 ppm of calcium. All this corn was really low in calcium. And some of the native corns tend to be that way. So blending it with corn grown in a different way would be a lot better from a nutritional standpoint. So with that, I know this was a bit of a whirlwind and there was a lot of extra stuff in there. It was pitched at kind of high level. So if you have any questions, please let me know.